All right, in this video, I'm going to be going over chapter 12 on the cell cycle. I'm definitely going to be emphasizing more on just the cell cycle itself, not so much mitosis. I'll try and post some video links to help you with mitosis itself, but the emphasis on this video is going to be the cell cycle and how that is controlled. So before we get going, you just want to kind of get a basic understanding and remind yourself of why cells would actually need to do any type of cell division. Key things are going to be for growth. Obviously, if you're growing, you need to replicate more cells. As cells start to differentiate or develop, you're obviously going to need more cells, as well as if you have some damaged cells have to be able to repair themselves. So typically this is showing you it's essential for those three components. But regardless of the cell or cell type we're looking at, all cells are going to come from some form of pre-existing cell. So mitosis, that's where you're going to be making an exact copy. In meiosis, you're still doing some form of cell division, but you're basically creating a half of genetic material. And for that one, you are typically just for sexual reproduction. But also, too, don't forget about prokaryotes. They reproduce by binary fission. So just remind yourself that it's prokaryotes. They don't do mitosis or meiosis. They just do binary fission. So moving forward, I just wanted to kind of show you this again. You've seen this quite a bit, but this is showing you a prokaryotic cell. You have your cell membrane. You also have, remember, the cell wall. So I'll just abbreviate that, the cell wall. Don't forget that prokaryotes have cell walls. This is showing you have the large circular chromosome. Remember, in prokaryotes, the DNA is circular. So you get that big, long, circular DNA right here. But all it does is it replicates the DNA. DNA gets replicated the same in all cells for the most part. Like helicase opens it up. DNA polymerase 3 adds the nucleotides the same. So you just want to remember that DNA replication occurs the same way. But once the DNA replicates, then the cell just divides in half and you get two identical daughter cells. So this is when you get those two that are identical, okay? Assuming no mutations take place. So just replicates, and then this happens really fast too. A lot of topics on the AP test will kind of get into, why do you typically see mutations more frequently in prokaryotes? It's just because they replicate so fast. That increases genetic diversity too. So this is looking at a eukaryotic chromosome. Oops. Now, a few things about eukaryotes in terms of the structure. It helps to kind of understand and revisit this. So if we had a chromosome, I would call this whole thing a chromosome. I'll draw this out. If there's my double helix, I would call this whole thing a chromosome. Now, if you looked at chromatin, I'll try and draw this down here. That's when it's unpacked. So for example, if we were doing a eukaryotic cell, and it kind of started to open back up here. Sorry. All right. So this region right here, chromatin, is when it's a lot looser. Okay. Typically, you only see DNA looking like this right before it's doing some form of cell division. Now, again, chromatin is when it's uncoiled. I also want to remind you guys that there is those little tiny histone proteins that get wrapped around. I mean, that's a little further down the road, but just understand that those little things are your histone proteins. These can be acetylated or adding a methyl group as well is going to help you read this gene. So understand that chromosome, chromatin, it has to just deal with the structure. So chromatin is when it's being opened up. We can kind of add to this is where if I were to read this, let's say that this thing right here is RNA polymerase. Your DNA is opened up. RNA polymerase could read that. And that's where I would actually read and make my mRNA sequence. So just understand that this would be where your mRNA would be read. So just kind of want to revisit the bigger picture. If this is the DNA, you have little sections of them, depending on what it is. Those sections that actually get transcribed, we're going to call a gene. So thinking bigger picture here. Now, if I were to kind of go back to this picture of these two drawings, right? I mentioned a second ago that I know that these two are going to be right before it does mitosis or meiosis for a couple of reasons. These two are going to be your sister chromatids. So basically, if you were to kind of think of it from a standpoint of the cell cycle, let's kind of try to put this into context here. Basically, if this is your DNA, then it replicates, then that's going to start to look like this. And again, it's only going to replicate when it's doing mitosis or meiosis. So I just wanted to put that out there and remind you guys that DNA doesn't typically look like this unless it's about to do mitosis or meiosis. So this right here is a full-fledged chromosome, but when it replicates, DNA is going to replicate, make an identical copy, and then it's held together at this region called the centromere. So this guy right here is your centromere, and that's what it's telling you here. So these two pieces are going to be 100% identical. So when you think about what is a chromatid, it's half of the duplicated chromosome. Well, remember, most of the time, this is, I'll put quotations around it, that this is when your DNA in a eukaryotic cell is normal, right? So that's the DNA normally. It's replicating because it's going to be doing mitosis or meiosis, then it looks like this. And that's when we call them a chromatid. So DNA is only referred to as a chromatid for a very small percentage of its lifetime, and that's just right before it's doing mitosis, okay? So just wanted to point that out there. Remind you guys DNA is made up of those nucleic acids, and there's multiple genes on them, but you just wanted to put out the basic structure there. So this is a screenshot of a biofix from Mastering Bio. I'm sure I'll post an assignment that'll just have the visuals for them. So just make sure you do watch those if you need those visual representations. So let's just take a quick look right here. So this is going to be, we're just going to focus on the left side for this video. So 2n equals 4. Just wanted to remind you that 2n, that 2 stands for you have 2 of each type, where n stands for the number of chromosome types. So in this case, it looks like they're referring to as length, that these two are homologous, these two are homologous. So the first thing that has to happen is the chromosomes are going to replicate. So this is your original parental cell right here. This is normal before it's doing anything. This is how the cell, for the most part, is going to be satisfied. Let's say it gets the signal to actually replicate its DNA, then you're going to replicate it, and now we can refer to them as having sister chromatids. So this 2n equals 4, that's what's referred to as that ploidy state. So it's 2n equals 4, 4 is your total number. So just understand that. So right here, my DNA replicates, it lines up at the metaphase plate, you pull them apart. The key thing is when you do mitosis, prophase, metaphase, anaphase, telophase, at the very end, it's still going to be 2n equals 4, 2n equals 4 in this example, because you want them to be identical. So that is critical to understanding what's the point of mitosis. It's to make an exact copy. Meiosis is going to be for chapter 13, so you want to take a look at that. Probably in the next week or so, we'll take more of a visitation on that, but this video is primarily just about mitosis. So in terms of mitosis, like I said, this is just kind of a brief overview. You're making 2 identical daughter cells. They're in your somatic cells, so those are essentially your body cells, which are also your non-sex cells. So basically anything besides your gametes are going to be somatic cells, right? This is just for humans, so we're 2n equals 46. Typically, assuming no mutations happen, there's going to be no variation if you're doing mitosis correctly, right? The DNA copies once, and that's going to be in the S phase of the cell cycle. So let's get into really what is the focus for this chapter. Besides the phases of mitosis, I just want to focus more on the cell cycle, okay? And before we get going, I just kind of want to reiterate, what is the point of these? What is the whole entire point of the cell cycle? It's really just to control cell division. So you need to understand cells just can't freely divide whenever they want to. It has to be a very controlled process for a number of reasons. You don't want to have errors in the DNA. You want to make sure cells have enough DNA. They're reproducing when they need to. So before we get into it, just think bigger picture. The cell cycle, the point is just to control it, okay? So let's take a look here at a few things before we get deep into this. I'm going to just remind you guys that if you look at this, what I'm highlighting all here, this is basically a time reference. So I think most of you guys are usually doing pretty good with this, but I just wanted to kind of remind you that what I've highlighted here, if this is like a representation of time, the majority of the cell's life is going to be spent in interphase. So you for sure want to just understand that it's okay. A lot of your cells aren't necessarily always going to be doing mitosis, and that's fine. You just want to remember, okay, a lot of stuff happens in interphase. When you look at the M phase, that's a lot shorter in relation to how long interphase takes, okay? So basically what happens in the G1 phase, that's for growth. So the cell is essentially growing, getting enzymes ready to actually replicate the DNA. That stands for the S is for your DNA is synthesized. Easy way to remember that. G2 is further growth. But these are your really two big key phases. You have the M phase and then interphase. Now within the M phase, you have two steps, which are going to be mitosis and cytokinesis. So it's kind of one of those weird things. Cytokinesis is not going to be grouped into mitosis. It's its own separate process. So key differences here before we move on is I want to point out, remember plant cells have a cell wall. So basically because they have that cell wall, they have to form a cell plate first. That's how they split their cytoplasm. Whereas animal cells, they kind of just like burrow in and it kind of looks like this cleavage furrow. And it just gets separated kind of down the middle, like tying a string around it and pulling it. That's how those get separated. So other than that, sound effects here. So I just want to remind you why the coordination of this matters. It's just about not all cells are going to have the exact same cell cycle. So you really just want to emphasize the point that cells need to know how and when to divide. So once we start to develop, certain cells are going to be stuck in what's called the G0 phase or G0. So it's all about coordination. So in terms of the frequency of cell division, it's not something that you guys need to know specifically, but it's just more interesting, right? So in the embryo, like as soon as the cells get fertilized, the sperm fertilizes the egg. They replicate about every 20 minutes. So that's a really, really fast process in terms of how quickly that they can divide. But as you start to develop, like your skin cells, that's one of the more active parts where mitosis occurs in us right now, but about every 12 hours. And you think about it, it makes sense, right? Because you damage those cells more frequently than anything else. So they replicate in about 24 hours, but other mature like nerve cells and muscle cells, they're going to be permanently in what's called the G0 phase. So they'll never actually get called back into the cell cycle. So the cells you have are the cells you have for your lifetime. So really interesting timing, but I think it's worth pointing out as we get into this. So in terms of this, let me turn down the sound volume on this. You want to understand, there's a couple of checkpoints here, but first and foremost, right, this is kind of interesting that if you look at this little animation, the DNA replicates here, and then you get your two chromosomes. These are different. You would call these at the ends, each one of these are going to be sister chromatids in relation to one another, okay? Then they divide. So let's kind of get into the checkpoints. The POGL did a good job with this one. They had those little flags. There's 3 of them. You have a checkpoint after G1. That's the first checkpoint that's going to check for growth. Do we have the enzymes? Are we ready to start to replicate the DNA? So we replicate the DNA and then we do growth again. This second checkpoint is going to check, can we do mitosis? And then this last checkpoint is really going to check, did we separate our chromosomes correctly? But really, really important for you guys to know that what is the point and what's the purpose of the checkpoints. So what is the point? It's basically to stop a cell from dividing that shouldn't be dividing. So when you think about it, very efficient in terms of how cells have evolved to do this. It's all about regulation. You want to make sure that as a cell replicates, it's replicating in the appropriate manner. You have all the energy needed. You're not going to have any errors. So putting it as simple as possible, the point of these checkpoints is to double check to make sure that we have everything we need to do division correctly. So The first G1 checkpoint, it's by far the most important in the sense that it's often the most failed. So typically, if it passes the G1 checkpoint, it's going to pass through everything. All we really need to focus on is that checkpoint right here. If I put that little flag for a checkpoint. If you look at what happens in the G1 checkpoint, this is a really nice overview. This is when you're synthesizing everything required for DNA synthesis. So now that we've kind of covered chapter 16 about all the enzymes, basically, this is where all these enzymes are getting built up, ready to do DNA synthesis. So once DNA synthesis is occurring, then it's going to grow some more. Typically, you're going to be building up additional proteins in the G2, and then you have your second checkpoint at the G2. So most of the time, if it doesn't meet the requirements, it gets kicked back into the G0 or G0 phase, but can get called back. So in terms of the major checkpoints, this is kind of the same thing, just emphasizing the point that it's all about that control. So in terms of the other, sorry about the sound, talking about the G0, like I said, it can be called back by external cues. Typically, a signal is going to call that cell from chapter 11 to start doing cell division. But a lot of them, like I mentioned, nerve and muscle cells earlier, they're going to be permanently stuck in that G0 phase. So not all cells can be called back. Now, let's think about this too. This is where things get a little more complicated. So what we were just going over with these checkpoints, we're going to call those internal regulations. So when we say that they're internal regulation, that basically means that the cell itself internally can control if it's going to be able to do mitosis or not. Another form of internal regulation is going to be this cyclin and cyclin-dependent kinases. So cyclin, this is great terminology here. So cyclin-dependent kinase, this right here is the enzyme. So this is your CDK. And the easiest way to remember this is you have to assume that the cells have these. Because you just have to make this assumption that the cell has an abundance of these CDKs. So, because enzymes aren't necessarily part of the reaction, so you just have to be aware that regardless of the cell has plenty of cyclin just there ready to be used, okay? Now, if you look at this, I have some phosphates here, so I would say that this protein is active. By adding these phosphates, you would say that this has now been phosphorylated and it's ready to be, it's turned on. So a couple of things that we need to know. It's not too terrible, but typically when we think of these internal versus external signals, it's all going to be based on how does cyclin bind to your CDK and then what goes on from there, okay? So, and this is just telling you the primary mechanism of control here, meaning how do these cyclins get turned on or turned off? Well, it's phosphorylation. So theoretically, if we were to kind of put this bigger picture here, let me try and erase this. Whoops. Let me erase this real quick. So if we're thinking from the last chapter, let's think of it from this way, where let's say that this is, I had a kinase here, and let's say I had a kinase enzyme with a phosphate, and that's going to basically turn on or activate this one. And then if you're thinking bigger picture, right, you could say that, okay, maybe there was a cell membrane here, there was a ligand that bound here, turn this one on, turn this one on. So this would be an example of, I'll abbreviate this GF for a growth factor. So when you think about it, you had a growth factor bound to this cell, activated this kinase, which is then activating this cyclin-dependent kinase. So basically, if you think of it from the last chapter, this is reception, this would be your transduction, and you're still in the transduction phase here when you're activating this kinase. So for you right now, you just need to know, okay, cyclin needs to get turned on. If you take a step backwards, well, how did this get activated? Well, it got activated by this kinase. How did this kinase get activated? Well, it was some signal, okay? So key thing, if you take a look at this, fixate on this picture real quick. If you look at this cyclin-dependent kinase, the terminology almost implies what it needs. It needs cyclin. So right here, Cyclin-dependent kinase is the enzyme. Cyclin is essentially the substrate that binds to it, right? So that's what you need to understand. Cyclin binds to the CDK, all right? Now, typically what you need to know is that when you have these two bound together, we can call that a mitosis promoting factor. So basically what we need to understand is that when cyclin binds to the CDK, you have a mitosis promoting factor, and that means that you're going to be doing mitosis. So basically you need to know the cell, remind you guys from that last slide, cyclin-dependent kinase, CDKs are always there, but the cell has to build up cyclin. So really there's a couple of things that we got to cover here to make this make some sense to us. So if you think about it, cyclin is what's called the activator. So cyclin right here needs to bind to your CDK. Now, the levels of cyclin in the cell are going to vary, so it just depends on where in the cell cycle we're at. CDKs, I'll say the concentration is always constant for the most part. And it's just easier to think of it in that concept, that if you have enzyme concentrations always the same. And now when you look at this phosphorylated cellular proteins, well, again, you have to activate this CDK. And if that's even too much, you at least just have to know cyclin binds to CDK. When these two are bound together, this whole thing, these two together, when they're phosphorylated, I can call that a mitosis promoting factor. That means you're going to be able to do mitosis. So in terms of this, let's kind of further this a little bit more. It's not too terrible, but again, cyclin has to be made in the cell. So think of it, cyclin is going to be the lowest in G1, and it's going to be the highest at G2. CDK, which again is your cyclin-dependent kinase, that's always constant. So basically when these two are bound together, you call that a mitosis promoting factor, okay? Now, again, when these two are together, that's going to allow it to pass through that checkpoint. So this is a really good graph, and I like this a lot because it shows you that at the beginning of G1, the cell gets the signal, okay, we're going to be doing mitosis. So you're basically going to start here. Your cyclin concentration is going up. Now, if I drew a line, and I've seen this on a few test questions, that line I just drew would basically be your CDK level, so your cyclin-dependent kinase. So just to kind of show you that CDK is always constant, cyclin gets built up. All the cyclin is going to bind to the CDK. When those two are bound together, that's when you have your mitosis promoting factor. So theoretically, if we were to take a look at this, to make this make some sense to everybody, when you think of it this way, if I have cyclin bound to what's called the CDK, one of them is not going to do much, but the more that you have, let's say I times that by 1,000, that's going to allow that concentration to actually allow it to pass through those checkpoints. So cyclin binding to CDK, that's going to be allowing it to pass through those checkpoints. So this is just kind of a nice overview of everything that's going on. If you can explain this, you're in good shape. So good little checkpoints here. Remember the key thing once you get to that M phase, that's where mitosis takes place, and then cytokinesis follows shortly after. Now, those two examples that I was just going through were going to be internal regulation, but we got to also talk about external regulation. So basically the stimulus to do mitosis comes from the outside. So I'll say comes from outside the cell. So a couple of things, growth factors are things like a hormone. So typically, if I had kind of like that previous example, if there's my signal, here's my receptor protein, it's going to bind here. And I'll say that this is kinase 1, kinase 2, and let's say that this is going to be cyclin. This is going to be basically activating cyclin. So basically, this is going to be my growth factor that's telling this cell to do mitosis. So that's an example of external, right? Now, density-dependent inhibition, pretty simple. So this one is kind of easy. Easy is a relative term, but basically if I have lost a chunk of these cells can physically detect that there is no more room. There's proteins that can detect that there's room, and so that's going to stimulate the ability for them to do mitosis. So there's some external cue to do mitosis. So that's going to be the stimulus from an external environment. Take a second and make sure that this makes sense. If you can explain these, this is all about the control of a cell cycle. So really, really helpful to understand the key components here. Like I said, the checkpoints, there's the three of them. You want to know those cyclin and those protein kinases. So cyclin and the CDK, and then 3 and 4 were those nice little kind of overview. Last one that we got to mention in this chapter, we'll do more when we get to the review of chapter 19 on eukaryotic gene expression. But basically, if you have a cancerous cell, that is some error of the cell cycle. So what is cancer in regards to the cell cycle? It means it's lost control of that cell cycle. So the checkpoints are damaged. So in terms of that, basically you want to understand proto-oncogenes normally activate cell division. So if it's switched on, we're going to call it an oncogene. So we also have tumor suppressor genes. So I'm going to hold off a lot of the detail on this one for right now. You just want to understand that there's a P53 gene that actually stops cells, and there's all kinds of images that we have talking about this. But for right now, I just want you guys to understand, here's a really good picture too, a better picture than what I was drawing, right? If here's cyclin is going to bind to your CDK when it gets phosphorylated. This is what we call your mitosis promoting factor. Pogel calls it a maturation promoting factor, but the same thing. Other than that, I think that's kind of a nice simple overview of chapter 12. Like I said, I'll post the links or an assignment on Mastering Bio for the mitosis components, but this is the main focus on chapter 12 on the cell cycle.